And our next speaker is Caitlyn Elliott. She worked in the phenotyping facility this summer, and she was mentored by Katie Murphy.
All right, good morning. I'm really excited to introduce Caitlyn Elliott. She is a rising junior at North Carolina State University. One major was not enough, so she's majoring in both plant biology and biochemistry. Caitlyn came to our group already with some research experience where she really focused on microbes that live in the soil. And so it was exciting to see her join our group and change that focus to really think about the plants and how the plants are changing with the environment. She contributed code to Plant CV. She learned not just one new language, but two in both Python and R. It's been such a pleasure to see her grow as a person and as a phenotyper. So welcome, Caitlyn. Thank you for all your hard work this summer.
Thank you so much for that introduction, Katie. My project this summer was on maize phenotypic responses to stress-conditioned soil. So for a little background, maize is vital to the US economy, food supply, and fuel. We produce 30% of the world's maize here in the US, mostly in the Midwest, which in 2024 looked like about $65 billion worth of maize, or about 15 billion bushels. I am less interested, though, in what we grew and more interested in what we lost. Also last year, we lost $1.3 billion worth of maize, or about 300 million bushels, to heat, drought, and wildfire. This may not seem like much of the total, given how much we grew, but what do those numbers actually look like? This is a semi-truck. Most maize in the US is transported in these. This is many semi-trucks and more semi-trucks, and finally, 300,000 semi-trucks, which is the number of truckloads needed to transport the maize that we did not harvest last year due to heat, drought, and wildfire. I'm going to tell you a similar story that you've gotten from others, which is that the climate is warming. Even moderate climate change predictions show increasing average summer temperatures, including across the US Corn Belt in the Midwest. So we must look for solutions to make our crops, including maize, more tolerant to the increasing threat of heat stress. My solution begins in the roots. All plants, including maize, release organic compounds into the soil. These are called exudates and can include amino acids, sugars, and secondary metabolites. These exudates allow the plant to cultivate their rhizosphere microbiome, or the microorganisms that live in the soil surrounding the roots. These microorganisms are important in supporting the health of the plant. A supportive microbiome can encourage the plant's growth and impact the above ground phenotype, showing a healthy, thriving plant. However, if the microbiome is not supportive, or perhaps a pathogen invades, we can see that reflected in the above ground phenotype as the plant appears less healthy. So how can we investigate the potential impacts of the soil on plant phenotype? Months before I came to the Danforth Center, individuals in my group conducted a heat stress experiment on two lines of maize, B73 and W605S. These are of interest to our group from prior work, and they grew these lines in control and heat conditions for 2 weeks at an early growth stage before moving them into control conditions to grow to maturity. At the conclusion of this experiment, they harvested all of the soil from these plants and saved it for me. When I arrived at the Danforth Center, I took this soil, for example, that conditioned by B73 maize grown under control conditions, and I planted a second generation of maize, B73, and grew it under control conditions in this conditioned soil. Additionally, I replicated the stress from the first generation experiment and grew B73 in this conditioned soil under heat stress. We also wanted to investigate if there was any impact of genotype on this conditioning, so perhaps growing maize of one genotype in soil conditioned by another genotype would change the phenotypic effect. So I also grew W605S in this conditioned soil under control and heat conditions. This is just an example of one of the crosses that I did. This summarizes all of the combinations that we did with 5 different soil types, including the crosses of those 2 genotypes and 2 temperature treatments, plus fresh soil as a control. Now that I have all of these plants and we are curious about the phenotypic effect or the effect on phenotype of this conditioned soil, how do I actually quantify that? I used Plant CV, introduced briefly by Oliver and Ennis as well. It is a free open source Python-based image analysis software developed at the Danforth Center by Noah Falgren and Malia Guillen. Using this software, I can analyze traits like height, color, chlorophyll index, NDVI, and more from images that I take of my plants. This allows us to see growth over time and quantify these traits that we observe. The first way that I used Plant CV was to process regular color images that I took of my maize at 2 different time points. Now, these images allowed me to quantify traits that I could see with my eyes, like height and color, in a more efficient way than I would have been able to do by hand using a Plant CV workflow. The first thing that we saw from this analysis was that B73 in fresh soil was affected by heat stress, particularly in its size. So here we have the genotypes along the top of the figure and the time points along the right side. The letters above the box plots indicate significance. So if 2 box plots do not share any letters, they're significant from each other. Here we can see that B73 grown in fresh soil was significantly taller under control conditions than under heat conditions. However, this significant effect on height was not reflected in other treatments of B73 or in treatments including W605S. Looking at another trait that we can observe from color images, we found that W605S conditioned soil affected color in W605S. Specifically, W605S heat conditioned soil had a significant impact on mean hue, decreasing the mean hue value under heat stress. That is highlighted in the green box. Mean hue is based on the 360-degree color wheel, where values around 80 to 100 degrees are green and lower values indicate warmer colors or more yellowing. So here, when we see a significant decrease in mean hue, what we're actually seeing is more yellowing of W605S heat-conditioned, W605S heat-stressed plants in W605S heat-conditioned soil. So interestingly, this was only reflected at the earlier time point from these images. When we analyzed the second time point a week later under heat stress, this difference in color was no longer significant. The second way that I used Plant CV to analyze my data was to look at multispectral images. Multispectral images taken with the crop reporter at the Danforth Center allow me to analyze traits that I cannot see with my eyes, and that includes things like chlorophyll index and FEFM. The first thing that we saw from this analysis was that W605S heat-conditioned soil affected chlorophyll index. Chlorophyll index is a measure of the amount of chlorophyll in the plant, and chlorophyll is a green photosynthetic pigment. So W605S heat-conditioned soil, which also negatively impacted mean hue from our previous analysis, negatively affected chlorophyll index, significantly decreasing it under heat stress. This was not reflected in the other soil genotype combinations that we tested in this analysis. Additionally, W605S heat-conditioned soil affected FEFM in W605S. So again, this is the same soil that has decreased mean hue and decreased the chlorophyll index value in W605S under heat stress. FEFM is a measure of photosynthetic efficiency in the plant, so lower values indicate less efficiency in energy production. Again, this was not reflected in other treatments that we looked at with this analysis. To summarize, heat had minimal impact on plant size. We did see that one effect, but otherwise it was not impacted across other treatments. W605S heat-conditioned soil caused yellowing in W605S under heat stress, and additionally negatively affected chlorophyll index and FEFM in W605S under heat stress. In the future, we would like to repeat this growth experiment, beginning with the conditioning process in that first generation of maize, harvesting the soil, and then growing a second generation of maize. In that repetition, we would like to test the conditioned soil for things like metabolites, microbes, and nutrients to understand what is happening in this conditioned soil that may be causing these phenotypic differences that we see. We'd also like to analyze the exudate profiles of the first and second generation of plants to understand the stress response of this maize further and see the differences between generations. I would like to acknowledge all of the REU staff, our funding sources, both those that came to us when we didn't have NSF funding and the NSF, the Data Science Core Facility for their assistance with Plant CV, and the Plant Growth Facility for keeping my maize alive while I was growing it. I would like to thank, of course, my lab and everybody in it for their support, including my mentor, Dr. Katie Murphy, and the summer intern cohort for their peer support throughout this whole summer. Thank you very much.
Questions?
I don't want to take anybody's slot.
But I have one. Okay. So I know you haven't done the experiment yet,
Yes.
but given your background in other research, do you care to speculate about what you think may have changed in the soil to influence the next generation?
Yeah, so interestingly, we actually saw a negative impact of this conditioned soil, which is not what we initially hypothesized. We thought, if anything, the stress on the first generation would cultivate a more supportive microbiome. So this begs the question, what then is happening in the soil? We don't know if it is something to do with the microbes in that environment or something else. Unfortunately, I am really not sure. We thought through, well, what nutrients are we giving the plant? We're starting with a soil mix that is regularly used in our experiments. So I would perhaps suggest that it is a microbe difference, but that's something we can really only know from repetition and further experiments.
Yeah, Kirk speaking. This is pretty cool. I guess the one thing that I'm wondering is there a shelf life to conditioned soil? Because you're growing these plants, and by the way, I don't think there's one magical factor, but if it's organic, I'd be worried about it being depleted. So I was just wondering, as you kind of go through microbes, I could see them kind of kicking around for a while. But do you have a sense of that? Like, could you leave it for a year and it would still have the same effect?
So in this experiment, it was left for a few weeks between harvest and when I grew it. And we did have questions if what is left in the soil will actually be able to stick around without carbon input for those couple of weeks. But this was a preliminary test to see, can we find any effects from this soil even being stored this way? And remember, in traditional agriculture, fields lay and fallow for months at a time. So whatever's being left in the soil is left for a long time, even though there's an active microbiome. Still, it is not having that same plant input until the following growth season. So while I'm sure there are some changes that occur over time, that might actually be more similar to what we would see in fields. Yeah.
Any more questions? Okay, if not, please join me in thanking Caitlyn for a great talk.